am really excited about these marks that we have identified. With respect to the exposome, it's been a bother. I follow a cohort of about 2,000 children, and at some point after these children were born, we did a random sample of about 100 when they were between, I'd say, 7 and 10 years old, and 10% of them had non-alcoholic fatty liver disease. I mean, a lot of fat in these children. So from that point, I decided to expand this research so that we could try and see whether we can find these marks that are established much earlier so that you actually can just not describe, but actually change the trajectory for some of these diseases. And this is the team that has actually worked on this for the most part. There is a very large field team that is following these children, and a molecular genetics person and a bioinformatics person that is supported by our center, and that is the core of the work that I'm going to talk about today. Then there are collaborators that range from social epidemiology to help us actually measure these social stressors, and people who measure the stresses, the chemical stressors. So what I'm going to do, I'm going to spend a very short time, because we've been talking about the challenges in the exposome research for a long time, all day long, and what I'm going to say is not going to be any different from what everybody else has said, and then really jump into this DNA methylation of these regions as response elements that are indelible, and you actually can retrieve them at any time during the life course, and be able to stratify populations that really do require interventions. And then I will show you the last part is the new array that has been developed by a company that develops arrays to actually start measuring this rapidly in all sorts of populations, so that we can begin to consider interventions. So some of these exposome problems that we're talking about include the fact that chemicals are measured individually, and we're exposed to a lot of them at the same time, and the fact that you have chemical and non-chemical stressors that co-occur, and it's very difficult to actually look at them in combination because our sample sizes tend to be small, and the fact that there is the external and the internal exposome that when you start combining them, usually it is very difficult to combine and measure and look at effects with outcomes. The other problems are the developmental window. It seems the developmental window is important, and this early developmental window seems like it's really what's going to shift. If we get the biggest bang for our buck, if we start early, and the Dutch famine study and the Chinese famine study showed us that. And also the fact that trying to measure exposure when we know that there are transgenerational effects and they're beginning to show up even in humans is usually just an exercise in futility, because all you can measure is what did your father smoke, which is what these people did, but you really don't know the totality of exposures that they were and the fact that the dose may matter in some cases is the study that was done in the Gambia. So beginning to think about really what are we talking about with this DNA methylation response at these regions. The question we want to answer is, if we identify patterns of environmentally responsive differential methylation patterns, can such patterns be used to infer past exposure and therefore be able to be used for risk stratification? And if you think about it that way, you have a whole lot of exposures here that we are exposed to, and this is not a complete, but what we think about is for each one of those exposures and in totality, the epigenome senses that exposure, and once it senses it, it nudges it in a way that is very specific, and that accumulated epigenetic nudging ends up contributing to disease. This is an example of how exposure to cadmium we measured we were able to identify these marks that are very specific to an environmental toxin that our population was exposed to. And we're able to identify these 1,900 regions in adults, and we measured them in children, and there was a major overlap of nearly 100 of them, suggesting that these marks are stable, a lot of them. So the question then becomes, why don't we just use these methylation marks the way they are? The problem is they do vary by cell type if you're just looking at anywhere in the genome. And also, when you are measuring them, you do not know whether these marks changed because of the disease or they changed in response to the exposure and they caused the disease. It's been very difficult to separate those types of things. And the other thing is that these methylation marks add, and the methyl groups themselves, they can be removed and added, and the stability is really not known. So the exception to this, there are parts of the genome that we can study, and this is where the imprint regulatory elements come in. These are DNA methylation marks that are used to control gene dosage. They go one after the other. They are very rich regions of these CG10 nucleotides, and because they control gene dosage, they're similar in every individual out there, right? And because they're established very early, they're similar also across tissues. And they are parent of origin dependent. So you have one of the parental alleles contributing, one of them usually silenced and the other one is working. So you know that when you measure methylation at places like that, in a normal person, it's going to be 50%. And to the degree that it varies from that means that you actually have a problem. The chemical and non-chemical stressors, though, they can curb this establishment of these methylation marks, and that's what we look for, whether they're chemical or non-chemical. This is an example of one of those regions that was looked at by the Dutch famine 70 years after. This was like 70 years after they had been exposed to the famine. People went back. If you look at this on the left-hand side in the red, what you're seeing are people that were exposed with this methylation profile was just dampened because they were exposed to the famine in the first trimester. And you look at the right-hand side, you do not see a problem like that. And afterwards, people have been looking at this in relation to diseases that people think, you know what, maybe I could use this as a biomarker so that we can risk stratify. So people have used it for colon cancer, people have looked at for in the Dutch famine, and there are some people that have looked at it in relation to cigarette smoke and as well as obesity in children. And this is just another example of this same marker being looked at in relation to maternal depression. Do you see the same type of dampening of these methylation marks when somebody has spent their prenatal period depressed? A similar mark is called the MEG3. And this is a region that is very similar to the one I just showed you. And it used to be called the adipocyte factor 1 because it is known to be causally related to obesity. So when we looked at, go back to look at the children I was talking about, you look at cadmium exposure, it was again related to changes, very big changes in methylation in these children. And then, but it was strange because when you did the GIS to see who exactly is exposed and does it coincide with where the cadmium exposure was, it seemed like it was only in the communities where ethnic minorities lived. So Hispanics and Blacks, 90% of the population lived in this community that was cadmium exposed. But for reasons that we are still trying to figure out, only the Black population seemed like it was, the effects were much stronger. And then you look at this MEG3, it has also been associated with other social stressors. It's not just the chemical stressors. It has been associated with maternal adverse childhood experiences. In fact, it's beginning to be thought of as a biomarker for people who are going to develop liver cancer. When these methylation marks are when they're very aberrantly methylated, they actually cause very, very serious diseases. But when they are not, when the damage is not as bad, usually what you see is something that is an aberrant methylation, it's not complete loss of methylation. Then you see an overrepresentation of neurological disorders, you start seeing metabolic diseases such as obesity, and this is where we found these children with liver diseases. And people are beginning to use it also to look at birth defects. But so far, all we knew was where these 25 regions are. They're really important. But we didn't even know where they were. So then we started looking for them. And this has taken maybe I would say the greater part of 5 years. And what the question we were asking is, where are the rest of these imprint control regions? Because if we can use them as biomarkers, they can actually be an exposure surrogate, so that by the time it's the kind of exposure, those that are responsive to specific patterns can then be used to predict who's going to be getting what disease. So where are these marks? And then is there evidence that these marks also, their difference from the 50%, which is what we call normal, associates with exposures that we know or outcomes that we know? Because if we can show that they're related to diseases we know or exposures that we know, then we can start thinking about them as risk markers. So we went out and looked for them, and the question we were asking was, if we do whole genome bisulfite sequencing, can we actually find them using the criteria that I was talking about, which is we are looking for places that are, we want to make sure that it represents all the germ layers, the endoderm, the mesoderm, the ectoderm, all of them, because we want to make sure they're similar all across the human body. And then you want to then look for them in the gametes, including the oocyte and the sperm, and then make sure that when you measure methylation, you are getting those at 50% everywhere. Where they appear at 50% should be a potential region of interest. Right? And so, yeah, this is the paper that shows that when we found them. So we had some that we knew, the 25, and we started comparing them to the ones we found using this algorithm that says, if you go into the genome, you find this area, it's 50% methylated, and then the paternal allele is 0, then the maternal allele is 100. So you go through the genome like that, and we found them. And there were 1,488 of them. And these have now been taken up by companies that really would like to develop them into biomarkers. So what we did was to pay this company to run the analysis and tell us first maternal arsenic exposure? Is it related to methylation at these regions in children, in the offspring? And we found yes. It seems 297 of these regions that I'm calling putative ICRs because some more experiments will need to be done. And then we asked the same question, which ones of these are associated with obesity in these children now that they are 4 to 5 years old? And we found, yes, there are 82. And among those, we find 31 of them actually are shared between the two. What that is saying is that, okay, if you were to go to a population at birth and look for these 31 marks, if we can repeat this, it would mean that we probably have found the markers we need that link arsenic to obesity from discarded tissues in umbilical cord blood, right? So what are we going to do with this information? I mean, at this point, it's actually not, a lot of people are beginning to use it, but at this point, it's geneticists. And the reason why I wanted to talk about it today was because this is a really good opportunity to think about how it could benefit your own research. If you think about, so the company is already measuring it in assisted reproduction because these are really easy things to measure. You get the samples and it's already documented what type of media you had, the birth outcomes that are documented in hospital and birth defects. But what they are not doing is looking for these slightly harder to measure exposures, the social stressors, the chemical stressors, and trying to figure out which ones of these can we come up with patterns that we can then use as marks that we can use for intervention. So the company now has actually compared it to the EPIC array version 2 that included some of these marks in their own array so that the built-in tests can be done as people conduct their research. And we are also continuing to work on this. But the bottom line here is it's really important to think of this in terms of looking for early prevention marks when you can still do something about it that is effective. And I'm going to stop here. Yeah.